Hello and good evening, everybody. On behalf of SigTuple, I want to extend a very warm welcome to all our audience today and another round of our fantastic series of education. And today we have none other than Dr. Thulasi Ramalingam, who is the consultant hematopathologist from Apollo Chennai. We are very happy on behalf of SigTuple to welcome him to be our guest speaker for today. SigTuple, as you know, is a company that engages with optics, robotics, and data science to create pertinent solutions in digital pathology. It's going to be a very invigorating, refreshing session today. So without any further ado, I would like to just give it over to Dr. Thulasi Raman. This is him, and you will be hearing him for the next 1 hour or probably a little more, as it's going to be a very interesting and detailed session on flow cytometry, compensation, and panel design. He has been the faculty member for national level cytometric conferences and an active member of international societies. He has played a pivotal role in developing and reporting advanced diagnostic assays for leukemias, lymphomas, plasma cell discrepancies, and a host of other hematological malignancies. His recent accolades include top honors at national conferences for his work. There is so much that we can tell about him. The list is endless. He's also been featured on the cover of the International Clinical Cytometry Journal. So please join me in giving a warm welcome to Dr. Thulasi. Dr. Thulasi, over to you, sir.
Yeah, thank you. Thank you for that introduction. And it's always a pleasure to talk on flow. And also, when you give a talk to an enthusiastic audience, it's always great. So without wasting time, because since it's a big, an elaborate topic, I'll just start sharing. So I'll just share my screen. I suppose you could able to see my screen, correct?
Yes, we can see you.
Right. So if you have any doubts, you can just post it in the chat box, or if you have any, when I ask for any comments or questions during the course of the lecture, you can, I suppose our organizers are kind enough to unmute you to interact for such questions and discussions, right?
Yeah,
So,
absolutely.
perfect. Thank you so much. So yeah, today it's a very, very important topic about flow cytometry, compensation and panel designing. Let's see how much we can cover. So I'll be finishing the compensation, and if we do have time, we can go to panel designing. So compensation is some topic, like most of us don't really understand what it is. Many people jump into reporting clinical flow cytometry, or even in research side, they start doing things, but they don't know some basic concepts, which is really important, especially something like compensation. Now, what is this compensation? So that is what the talk is all about. Now, so this is the principle of flow cytometry. So what does flow do? Flow basically can characterize any cell based on its physical properties, which is the scatter, forward scatter and side scatter. And beyond that, it can also characterize cells based on the proteins or antigens which is present on them. Now, just to make things clear, with this is a small schema where you have a cell which has to be studied, and this is an antigen which is unique. Let's assume that this is unique to this particular cell. Okay, let's take this as a B cell, and this is CD20 antigen. Now, what is the principle of flow? You add a complementary antibody for this CD20 molecule, which is tagged with the fluorochrome, right? So when you add, what happens? It goes and binds to the cell. Now, this cell, which is tagged with this antibody tagged with the fluorochrome, is passed into a flow cytometer. The fluidics and everything make sure the cell passes in a single file pattern, which we call it as hydrodynamic focusing. Then as it passes through in a one-by-one fashion, which is we call as the single file pattern, there is a laser, which emits a monochromatic light. So when you come to the basics of flow, you should know some amount of physics. Okay. So what is a monochromatic light? Anyone want to try? Is it possible? Like you can, monochromatic is something like a laser emits light of monochromatic, which is it emits light of single wavelength, specific wavelength. For example, if it's a blue laser, it emits blue light of wavelength 488 nanometer, right? So it emits a light of single wavelength, which hits the fluorochrome. Now, what happens to the fluorochrome? The fluorochrome gets excited, right? It goes to an excited state, and then it comes to the ground state, releasing light of lower energy and longer wavelength. Okay, and such a light will be captured by the detector. Now, if this particular cell has CD20, this antibody tagged with fluorochrome will be bound to the cell and you will get the signal. If say if it's a T cell and it doesn't have the CD20 molecule, this antibody will not bind and you will not get the signal. So that's how we discriminate cells by flow, right? And remember that this is just a schema where I showed you one antibody, but in real-time flow cytometry, we stain the cells with multiple antibodies, right? Multiple fluorochromes based on the configuration of your instrument. For example, if you're using a 20 color flow cytometer, now I add 20 different fluorochromes simultaneously at a single point. Okay, and all get excited and they are captured by the detectors and you get the signal. Now, what is compensation? Let's go back. So this is just to show you the fluorochrome again. I'm just going to show you this particular molecule now. So this is the fluorochrome and this is the incident light, okay, which is coming from the laser. In the flow cytometer, it goes to an excited state and it cannot stay there for a longer time. Then it starts to decay, which means it comes to the ground state. Now, while coming to the ground state, it releases energy, releases light of a longer wavelength and lower energy, right? This is what a fluorochrome mean, right? Now, when we talk about this emission of light, so it absorbs light from the laser, which is monochromatic. I repeat, it absorbs light from the laser, which is monochromatic, a single wavelength light, and emits light at a different wavelength, okay, especially a longer wavelength and lower energy. Now, this emitted light, you need to remember, is not like a monochromatic as the incident light, right? It is a spectrum, which means now you take the most common fluorochrome, what we talk is FITC. I suppose those who are working with flow or know something about flow should know about this fluorochrome, fluorescent isothiocyanate, which is FITC. Now, this is the laser line, okay? This is the blue laser, which emits a monochromatic light of 488 nanometer. So at this particular wavelength, the FITC get excited, okay, and it emits light of a different wavelength, right? This emitted light is called emission spectrum because it is not one single wavelength. It is a spectrum of light which is getting liberated from the FITC molecule. So for example, if I would say 480 to 650 nanometer is the spectrum of light which is getting released by FITC. Okay, so this is called emission spectrum. Emission spectrum. Okay, so this emission spectrum is very important. So I repeat, any fluorochrome, when it is emitting light, it emits light as a spectrum, not like a monochromatic as your laser. It emits light in a spectrum of wavelength. For FITC, it's around say 480 nanometer to 650 nanometer. So what's the problem with that? Now assume that there are two fluorochromes. So we just spoke about FITC. Now in my experiment, I'm using two fluorochromes. Let's assume FITC and PE. For example, if I want to study the B and T lymphocytes, now I use CD20 FITC and CD3 PE. Okay, and same, the cells are stained and they are going into the flow. Now, FITC gives you an emission spectrum, correct? Which is fluorochrome A. Assume that fluorochrome A is your FITC. It gives an emission spectrum like this. And P is another fluorochrome, which gives an emission spectrum, something like this. For example, yeah. So for example, 400 to 500, and this is some 450 to 600. Now, at one point, you can see there is a overlap in the emission spectrum of two fluorochromes, FITC and PE, right? This is called as spectral overlap. This is called as spectral overlap, okay? So what is the importance of the spectral overlap? We'll see. So you can see here, this is again, I'm showing you, this is FITC, which is giving a green light, which is a wavelength between 480 to the emission spectrum of FITC is from 480 nanometer to 16 nanometer. And the emission spectrum of another fluorochrome, which is P, which is, mind you, this is also excited by blue laser. Okay, so this P emission spectrum is from 550 nanometer to say somewhere around 700 nanometer. And at one point, for example, say 550 to 600, you have an overlap. This is called as spectral overlap between two fluorochromes. This is called as spectral overlap. So what is the problem with that? What is the problem with that? The problem is that
Dr. Thulasi, there is a question in the chat box.
Yes.
By Dr. Shanti. She's
Yes.
saying emission spectrum entirely depends on fluorochrome and the antibodies. Do they have any role in it? Do the antibodies have any role in it?
Antibodies have no role in it. Emission spectrum is completely a property of the fluorochrome. See, the fluorochrome is like a tracer molecule. It is like a label. You can tag it with any antibody, irrespective. You can tag it with any antibody. So it has the antibody per se. For example, this anti-CD3, anti-CD19, anti-CD20 has nothing to do with emission spectrum. Emission spectrum is fully a property of the fluorochrome, what we are talking. Okay. And when you talk about spectral overlap, you are talking about multiple fluorochromes, minimum two to maximum any number based on the numbers what you use. Yeah, I suppose that is answered, right? Fine. Now, so this is spectral overlap. Now, what is the importance of learning the spectral overlap? I'll tell you. Now, how these emitted light are, once the fluorochrome emits light, right, the emission spectrum, it is captured by a detector, obviously, as I showed you in the schema, the earlier slide. So now this is the emission spectrum of FITC, right? And this is the emission spectrum of P. And as I said to you, this is the spectral overlap. Now, what is the problem? You can see that a part of FITC light, the emitted FITC light or the emission spectrum of FITC is getting into the PE detector. So this is the bandpass filter of PE, which allows light between a nanometer of 580 to 600. Okay, and you can see a part of FITC light which falls in the range of 580 to 600 is getting into the PE detector. So ideally speaking, theoretically, the PE detector should receive light only from the PE fluorochrome. Correct? The light emitted from the PE fluorochrome should go into the PE detector. Light emitted from the FITC fluorochrome should go into the FITC detector. But you can see, since there is a spectral overlap, you can see a part of FITC light is getting into the PE detector. Now what happens? The detector will take this as a PE signal, but actually it is not. It is the FITC light which is getting into the PE detector. There comes the importance of spectral overlap. And that is the reason why we need to have an idea about the fluorochromes and the spectral overlap and how to combat it. Right. So every fluorescent molecule can emit light with a spectrum. Okay. Remember, all the fluorescent molecules cannot release monochromatic light. They emit light in a spectrum, and that is called as emission spectrum. Okay. And when the emission spectrum of two fluorochromes overlap, it is called as spectral overlap. Right. So because of this overlap, some amount of FITC light for the see, I'm just using FITC and P because they are the more common fluorochromes. It can happen with any two fluorochromes. Okay. Some amount of FITC fluorescent can get into the other detectors. For example, FITC, the very close, the nearby channel is P. So some amount of FITC light goes into the P detector. Okay. And it is recorded as P signal, which is false. Okay. And this term of spilling over of signal of one fluorochrome into other channel is called as fluorescent spillover. This is called as fluorescence spillover. Okay. So the spilling over of FITC signal into the PE channel is called as fluorescent spillover. Is that clear till now? We have all the time. We can go slow. If you have doubts, you can ask me. You can put it in the chat box. I can take it between, because this is a complex topic. If I go fast, then there may be some difficulty. So you can, I suppose, Dr. Renu can stop at intervals and can put the questions. Is that fine, Dr. Renu?
Yeah, that's fine.
Perfect. So now you see, I'm just showing you as a data. Now here, I'm just staining the cells only with FITC, correct? I'm staining the cells only with the FITC. I'm not used any other fluorochromes. But when I run it in the flow cytometer, you can see this is the emission spectrum of FITC. So actually, this is the FITC channel, which detects light of FITC, which is ideal, correct? But you can see that some amount of FITC light is also entering into the adjacent detector, which is PE detector, right? This is called spillover. Now, how does it look in the real-time data? You can see this is actual correct signal. See the FITC, you have stained the cells only with FITC. You can see the FITC signal, perfect. But here in PE, I have not stained the cells with any PE fluorochrome, but still you are getting a signal. This is noise, this is negative, perfect. But why should I get this? Because I've not added PE, I've not stained any cells with PE, I've not used PE fluorochrome in the experiment, then why should I get a signal when I used only FITC? That is because some FITC signal is spilling over into the PE channel or the PE detector, right? And when you see the data in histogram, it appears like this. When you see it as a dot plot with a 2 parameter, you can see this, right? This is called spillover. I suppose everyone is clear with spillover now, right? Correct. So everybody knows what is spectral overlap and everybody knows what is spillover, right? Now, I'm going to repeat again and again until the concepts are clear. So you see this is FITC emission spectrum, and this is PE emission spectrum. And you can see this is the detector with a bandpass filter, which allows light between 530 plus or minus 30, which means it will allow light between 500 to 560 nanometer to go and hit the FITC detector. Similarly, you have this PE channel with the PE detector, which allows light with a bandpass filter, which allows light between 550 to 600 to pass through it. Now you can see that this FITC emission is a spectrum. So all this light is captured, which is very good. But you can see a proportion of FITC emission spectrum light is getting into the PE detector, right? This part. This is a FITC spillover into PE. Similarly, if you have a close look, some amount of PE emission spectrum, very small, is spilling over into the PE detector. This is PE spillover into the FITC detector or FITC channel, right? So if I highlight you can see that this is the FITC spillover into PE, quite significant. And here you can see a small PE signal spilling over into FITC channel, right? If you see the percentage in this example, 22.7% of FITC light is getting into PE. But a very small proportion, 0.7% of PE light is spilling over into the FITC channel, right? Now, what to do with this? So this is something which we don't want. We don't want a false signal in PE, which is coming from FITC. We want, ideally speaking, what we want? We want only the PE channel to detect only the PE signal, purely the PE signal. Similarly, we want the FITC channel to detect only the PE signal. But here there is a crossover. There is some spillovers. So how to do this? So basically, we need to eliminate this FITC signal from the PE detector, correct? We need to eliminate this FITC signal from the PE detector. Similarly, we need to eliminate this small PE signal from the FITC detector, right? So that is called as compensation. So compensation refers to correcting a phenomena called fluorescence spillover in flow cytometry. This is removal of a signal of any given fluorochrome. For example, let's take FITC. So removal of FITC signal, okay, from all the detectors. Okay, I'm just giving you an example PE, but if you're using a 10 color instrument, you have, including FITC, you have 10 detectors, right? So when you run only, when you're talking about FITC spillover, except FITC detector, if the signal is falling in all other detectors, that has to be removed. Okay, this is called as compensation. So it is removal of signal of any given fluorochrome from all detectors except the one dedicated for it. So if it's a FITC fluorochrome, you have to remove FITC signal from all the detectors except FITC detector or FITC channel, right? That is called as compensation. Now, remember why this compensation topic is very important, because I have seen a lot of flow cytometrists, clinical flow cytometrists who do leukemia, lymphoma, myeloma, MRD, and so. When they send me the data, sometimes the data looks so bad because of improper compensation. And mark my words, like if you are not thorough with your basics, you will be going for a ride. Because let's assume that some population is giving a false signal for a important marker with which you're going to call as a disease. Now, this is maybe because of some improper compensation. It may be a false signal. Now you will be happily calling that a diagnosis of so-and-so, right? Actually, it's not. You're giving a false diagnosis. Just interpreting the plots, it may apparently look that the data is showing abnormal, right? Maybe like assume it's a BLL or something. But if you check the compensation and if you correct the compensation, it may be a normal one. Okay, because I've seen, because many plots comes to me for opinions and so on. So many times this compensation is not well done, and because of that they land up in problems, right? So this is very poorly understood, and you should really know about this before doing any complex experiments. Now, coming back to this particular example, now let's do a manual compensation for this. So here you saw that there is a 22.7% of FITC signal spilling over into the PE detector and some 0.7% of PE signal. When I say signal, it's the emitted light or the emission spectrum. Remember, 0.7% of PE light is spilling over into the FITC detector or FITC channel, right? Now, how to remove this? Ideally speaking, so this is FITC, green light, and you can see that 0.7% of FITC, red light, coming into the FITC channel or FITC detector. Similarly, here you can find that almost like 87% of PE light is getting into the PE detector, which is very good, correct? But you can also see that 13% of green FITC light is getting into the PE detector, which is not needed, which we have to remove. So ideally, you will be removing this, right? To compensate, you will be removing this. Okay, so this is called compensation. Okay, now this compensation can be done manually, but now the existing instruments have automated protocol where they can do compensation automatically, right? I'll show you how it is done, right? So this is the spillover table. So this table is called spillover table. You can see that, see, 100% of FITC is going into FITC, which is very good, but you can see that 22% of FITC light is also going into PE. 22% of PE light is getting into PE, which is not good. And some amount of light is going to the other detectors also. Similarly, in PE, 100% of PE goes into PE, but there is some spillover into FITC, right? So when you make a table or a matrix like this, that is called as spillover matrix. Basically, you are getting the percentages of spillover here. Simple. So if you see check diagonally, it should be 100%. And the spillovers come here. So whatever I have highlighted is green is green light spilling over into other detectors. Now this is called as spillover matrix. Okay. Now the inverse of this, okay, that is 1 by spillover matrix or spillover matrix minus 1 is called as compensation matrix. So this is compensation matrix. So whenever you see your instrument, there will be a compensation tab. When you click, a table like this will open. This is called as compensation matrix, which will tell you the spillover details and the correction details, right? This is called compensation matrix. So whenever you do a compensation exercise, you will be checking this, right? This is very important, right? So before I go further, any doubts, Dr. Renu? We have any questions?
Not yet. Nothing.
Perfect. So I suppose people are on track. Great.
Yeah. If anybody has a question, you can also on the React box, there's a heart symbol there. You can even just raise your hand. So, that way we know that you have a question.
Or if you understood also, you can click a thumbs up sign so that I know that you people are there with the concept. Okay. When I ask you, it's all okay. You can just put your thumbs up also. Great. So, any doubts or everything is okay? I suppose everything is okay. Great.
Okay.
Right. Now my question.
Up. There you got the thumbs
up. Right. Great. So now the question is, now, is there a way you can avoid compensation totally? Can anyone reply? Or you can unmute and answer that. Do we have the facility for that, Dr. Renu? They can unmute themselves?
Yes, they can. Yes.
Yeah, then we can be interactive. Thank you. That will be interactive. Come on. So is there a way you can avoid compensation? You can unmute and you can answer. Come on. Let's be interactive. This topic needs a lot of interaction.
I'm going to just say one thing here. The person who's the most interactive in this whole session will get a special prize on behalf of SigTuple for being the most interactive person in this session. And Dr. Thulasi, you can have one winning question at the end of the session. And we'll see who answers that. They will also get a prize.
Great. That's nice. Great. Yeah. Yes, now. So you have a reward here. Come on. So is there a way you can avoid compensation totally? So compensation problem comes when you're using many fluorochromes, right? So is there a way you can avoid compensation totally? What are the ways you can avoid a compensation? Yeah.
Hi, sir. Vishesh here. There are certain ways we can reduce the spillover, but not totally avoid considering the traditional flow cytometry. One way is to titrate antibodies before using them.
Okay.
Another is during instrument setup, we can do voltration.
Okay. Yeah, Vishesh, I really agree with your points, but to make it very simple, like for beginners. Panel designing. So what all you need to consider, say, for example, you have a 10 color flow cytometer. Okay. And now your experiment has 3 fluorochromes. Okay. Now, is there a way you can completely avoid compensation?
Yeah, you can select the fluorochromes and primary channels.
Okay.
For example, FITC, APC, and BB421.
Yeah. So basically, what he says is you can select the fluorochromes where there is no spectral overlap. Correct.
Yeah.
When you're selecting
Right.
fluorochromes with no spectral overlap, obviously you don't need to do compensation because there's no overlap. Yeah. So any other ways of doing it?
There's a chat box comment by Dr. Gautam
Yeah.
Bhagat.
Yeah.
Titration. If we save pre-saved comp fills, then we can avoid for further case analysis. That's
Yeah.
what you
So
said.
titration is something like you need to remember that it's related to the volume of the antibody. Still, there is a spectral overlap happening. Okay. Remember, titration is only setting the amount of volume antibody to be used. For that, you don't have a background noise, background signal, right? Dr. Rasha is there. So Rasha, she wants to try. Is there any other way you can avoid compensation? You can unmute and you can answer. Yeah.
Yes, we can use the laser delay to eliminate the
Yeah.
spectral overlap.
Perfect. So other thing is you can distribute the fluorochromes across different lasers. So in that way, you can avoid compensation because you can use the gain of laser delay, right? I'll show you that. Now, see,
So we also have Lena on the chat who's saying choice of fluorochrome.
Yeah, choice of fluorochrome, obviously.
And Dr. Satya was saying we can do by selecting the markers based on the expression.
Markers has nothing. Again, see, antibodies don't bring antibodies. Antibodies, compensation, spectral overlap is nothing to do with the antibodies, as I said you earlier, right? Now, you see here, now, if I use FITC and P, for example, now I have a two-color experiment, simple experiment, two-color experiment. If I use FITC and P, obviously there is a spectral overlap, right? I need to compensate. Now, since it's a two-color experiment and I have a five-color instrument, I can spread it across FITC and another fluorochrome. For example, PC7 or something, where I don't have any spectral overlap at all. You see the emission spectrum of FITC is here. The emission spectrum of the other fluorochrome is here. I don't have any spectral overlap. So obviously I will not get any spillover and I don't need to do compensation. The other way is around, like Dr. Rasha said, though there is a spectral overlap, the emission spectral overlap between PB and FITC, which is Pacific blue, and FITC. Now, Pacific blue is excited by violet laser, whereas FITC is excited by the blue laser. So with the help of laser delay, we can eliminate the spillover and compensation issues, right? So this is a second way of doing it. Okay, so basically you can do this, but is it worth doing? See, you have a 10-color instrument. Okay, what would you try to do ideally? You want to do an experiment with 10 fluorochromes. That is why you bought a 10-color instrument. What's the point of doing a 2-color, 3-color instrument in a 10-color mission? You are underusing the resource, right? So though compensation is inevitable in multicolor flow cytometry, so if you want to use your instrument to its full potential, you should know about compensation. So afraid of compensation, you cannot underuse your instrument, right? So ideally, you need to know about these things. So one thing you need to remember is normally spillovers happen between adjacent fluorochromes, adjacent fluorochrome channels. For example, FITC-PE. Okay, FITC-PE, there is a spectral overlap, means spillover happening. Similarly, you can take the tandems, closed tandem dice. I suppose everyone knows what is tandem dice. So take tandems, parent and the tandem dice. P will commonly spill over into its tandems, like P Psi5 or P Psi7. So spillover normally across the adjacent channels. So I can show you this beautiful spillover table in a thing where you can see it's a 10-color instrument, which is of a Navios instrument of Beckman-Coulter. You can see FITC spills into FL2, which is your PE channel, and to the third channel, which is ECD, which is PE Texas red, right? Similarly, if you see PE doesn't spill over into FITC, but it spills over into its tandems, which is PE Texas red, ECD, what people call, and then it spills into PE Psi5 and PE Psi7. Similarly, if you take ECD, it spills into the adjacent channels. So spilling over across a distant channel is less possible, but yes, it can happen. But normally spillover happens between the adjacent channels because of the significant overlap spectral overlap between those two, right? Okay, so I suppose no doubt still now, right? So we are thorough with what is spectral overlap, what is spillover, and what is compensation. Okay, now let's check into the experiment to do compensation, because you cannot avoid compensation. Obviously, you need to compensate for your instrument, do compensation for instrument. Now, how will you do it? First, anybody want to try? You can try, put your hands up. How will you compensate for your experiment? Anybody want to try?
You can go ahead and give your answers. Don't worry, we're all learning here. So
Yeah.
even if it is not the
Yeah,
right way, we're all here to learn. There
Yeah,
we
you
go.
can. Yeah,
From
exactly.
Senthil K using single stain.
Single stain. Very good. Perfect. Bang on. Single stain. So what is single stain? Anybody want to try? What is single stain? Yes, Dr. Vishesh, you want to try what is single stain? Right.
Yeah.
So how
it's
yeah.
basically, sorry for delay. So basically we can prepare samples with similar autofluorescence and stain with selected fluorochromes.
Okay.
Like for example, if they are PBMCs, then we can stain them with CD4 FITC, CD4 PE, CD4 EPC, like that.
Right.
And create
So
controls.
agreed. So what is single stain is, for example, now you are using a 5-color instrument. So it has FITC channel, it has PE channel, it has ECD channel, which is a third fluorochrome, which it has PC5 channel, PC7 channel. Okay, there are 5 channels. Now what is single stain is, now I'm going to take a sample, stain only with FITC, only adding CD FITC, FITC molecule. Okay. Now, ideally, if I run it in the machine, where should I get the signal? I should get signal only in FITC. That is correct. Now I'll be looking into other detectors other than FITC, like PE, ECD, PC 5.5 and PC 7, whether any signal is coming. Ideally, if there is no spillover, there will not be any signal in other channels. If I get signals in PE, ECD, PC5 and PC7, that means there is a spillover of FITC into those detectors. So that is the best way to know whether there is a spillover happening. First, this is called single stain, running a single stain control. Second, once you checked that there is a spillover happening in other channels, now you need to do compensation, which is removing that spillover signal of FITC into other detectors, from the other detectors. That is called compensation. Now this compensation can be done by manually or by automatic methods, which is available with the instrument itself. So once you have done with FITC, now is that over? No. Now you need to run cells stained only with PE. Now what you do, you look for signal in PE, which is correct. Now you need to look for signal in FITC, ECD, PC 5.5 and PC 7. If any of the signal now running the PE tube you get in other channels other than PE, that is spillover of PE into other detectors. Similarly, you need to eliminate them, remove them. Then you need to run for the third fluorochrome, ECD. Check for signals in other detectors. Then you run for cells stained only with PC 5.5, run it, check for signals in FITC, PE, ECD and PC7. If there is any signal coming there, that means it's a spillover, you need to remove them. So you need to run cells stained with one particular fluorochrome, which is called single stain, for every fluorochrome to check for spillover and correcting them. Okay, so that is how you do single stain control to compensate. Now I'll show you here. See, I'm using a 3-color panel here. Okay, so I'm using FITC, PE and propydium iodide, which is the third fluorochrome. Okay, now first I'll run FITC. I'll check whether any signal is coming in PE and PI. If there is signal in PE and PI, it means FITC spilling over into PE and PI channel. I need to eliminate them. Then I run cells stained only with PE. Now I check for FITC. If there is signal of PE spilling over into FITC, I need to remove them. Similarly, if any signal is coming in PI, I need to remove, because this is only stained with PE. It should not ideally show any signal for FITC or PI. Okay. Finally, I run the third tube, PI, which is propydium iodide. Okay, cell stained with propydium iodide. When I run, ideally it should give signal only in PI channel, which is for PI. If any signal comes in FITC and PE, it has to be removed. And that's how you compensate. Okay, you run single stain, run it, check any signal is there in other detectors apart from the one which is designated for it, and you need to remove it manually or by automatic way from the other detectors. That's how you do compensation. Okay. Now you see here, this is, I've run a single stain FITC. Remember, I have run a single stain FITC. Now you can see that there is a signal in PE, which means, is it true signal or false signal? It's a false signal because I've stained only with FITC. How come you can get PE signal? You should not get, right? So now if I give automatic method to compensate, this compensation will be done. And now it is compensation. So the FITC signal from PE is been removed and you get FITC fluorochrome light detected only in the FITC channel and not in any other data, especially in the PE detector. Okay, that's how you do compensation. Okay, so for that you need to run the single stain controls. Now, what are the single stain controls? So for preparing a single stain control, so let's assume, so now to do the single stain control, can you just run fluorochromes into the machine? Practically not possible, right? The fluorochrome has to bind to an antibody. The antibody has to bind to a particle, which could be a cell or a bead. Then only you can run it in the flow cytometer. Though spillover compensation is all related to the fluorochrome, you cannot just run the fluorochrome in the machine, right? It's just a tracer molecule. You cannot run them. So the fluorochrome has to be bound to an antibody and it has to be bound to a particle, which has to go in through the flow cytometer. So it could be the particle could be a cell or it could be a bead. Bead is a fluorosphere. Okay, it could be anything of this. So you can use biological cells or beads and antibody to do the single stain controls. Okay. So now what you do, as I said, you fluorochromes cannot be directly sent into the flow cytometer. It has to bound to an antibody and this antibody has to bind to a particle like a cell or a bead. So what you do is that, and there are certain important points which you need to consider when you're taking a single stain control, a cell or a bead, whatever. You need to consider these points. They should have a well-defined negative and a positive population. Okay, so when you are using beads or cells as a single stain control, they should give you a negative signal as well as a positive signal. And the positive signal should be in the brightest level. It should not be weak, because when you compensate with a brighter signal, during your experiment, if you get bright signals, that may not be adequate enough to compensate for it. So it will be giving a spillover. So when you're using a bead or a cell, you should have a negative and a positive signal, and the positive signal should be brightest in the single stain control. Okay, and you should use fluorochromes which you use in your experiment. For example, I'll tell you this one I just encountered. I'm using FITC in my experiment. Okay, you cannot use Alexa Fluor 488, which has same emission and excitation as FITC. You cannot replace them. If you're using FITC, you should use single stain for FITC only. You should not use it for other fluorochrome. Okay, and on the other hand, so as I said you, the single stain controls can be cells or beads. So what is the advantage of bead? What is the advantage of cells? Okay, now you need to think about it. So for example, if it's a bead, there is something called commercially available as compensatory beads, which we call it as comp beads. Now, the advantage of these beads is that, you see, this is the comp bead. It has anti-kappa molecules on its surface. I repeat, these beads have anti-kappa molecules on its surface. So you add any antibody tagged with any fluorochrome, it goes and binds to it, because every antibody has kappa light chain on it. Antibody means it should have a heavy chain and light chain, right? And all antibodies what you're using in your laboratory, let it be CD19, 27, 54, whatever, they have kappa light chain on them. And these beads are designed in such a way that they have anti-kappa molecules on its surface. So when you add this antibodies, it will go and bind. Okay. So on the other hand, if you're taking cells, that's more tricky, because if you take lymphocytes, you should use antibodies tagged with that respective fluorochrome, which you want to check for compensation, whether it will go and bind to the lymphocytes. That's the problem. If you use beads, you can use any antibody, it will go and bind, right? So I'll give you an example to make things clear. For example, in your lab, you are having CD123 FITC, and now you don't have any other FITC marker, antibody in your lab. You have only CD123 FITC. Now, when you want to use that for doing a single cell control, can you add CD123 in your peripheral blood? it will not bind to the lymphocytes, right? So you cannot use that to set your experiment for a single stain control for FITC, right? But when you use beads, that 123 is an antibody, obviously. CD123 is an antibody, and it has kappa light chain on it. And when you add it to the beads, these anti-kappa molecules on its surface will bind to that antibody. Now, no matter it is CD123 or any antibody, you can add them, it will bind to the beads. So that is the advantage you have with beads compared to cells. Okay. So which one you prefer to do single stain controls, whether beads or cells? So as I said, beads, very easy to use. So for example, for rare antibodies, you can just add, it will bind, right? And you can use it with any antigen. For cells, it should be an antigen, as Dr. Vishesh said, it should be an antigen which is expressed on the lymphocytes, because we use lymphocytes as the important cells because they don't show autofluorescence. So we use lymphocytes to do for compensation single stain control experiment, we use lymphocytes. But you should have all the antibodies which can bind to the lymphocytes in different fluorochromes which you want to compensate. That many labs may not have. If it's a small lab, they may not have all the lymphocyte markers with respective fluorochromes, FITC, PE. I'll show you how I do it in my lab. But if you're a small lab and you don't have that array of antibodies, then you can use beads, which is very easy. No matter of what marker it is, you can just select the fluorochrome. FITC, let it be any rare antibody tagged to FITC, it's okay. You take FITC. Similarly, PE, no problem with antibody. You just take PE. You can just take, because they're all antibodies, and when you add it to the beads, they will bind. Whereas in cells, you need to check whether it will bind to the lymphocyte. Right? So, but the problem with beads is that you are working with cells. We are working predominantly on cells, so beads may not be a perfect match. Okay, but they give a beautiful separation between the negative and positive signals, and they'll also give you a bright signal. So that will be very ideal to compensate. Cells may not give you such great separation between the negative and positive, because let's take CD4. Now, not CD4, let's take CD25. If you have a fluorochrome with CD25, right? Now, CD25 is expressed in activated lymphocytes, but the signal may not be that great. It will be falling very near to the negative signal. Okay, so that may not be great. So when you're selecting beads, you get this advantage of good separation between negative and positive population, which you may not be able to get in cells when the markers are not quite correct, right? But when you take cells, the advantage is it's a better match, because you every day work with cells, biological samples. So they have a better match with the experiment, what you're doing. And the problem with cells is that you need to lyse the RBCs, because when you take peripheral blood, you cannot directly use it. You need to lyse the RBC. So it's going to take that extra time and lysing step extra. And finding, this is what I was telling, specimen for each type of antigen could be very difficult, right? Because you may not have fluorochromes with antibodies that will bind to lymphocyte, right? You may have CD15 in one fluorochrome, for example, P. Now 15 will not bind to lymphocytes. So you cannot use that P CD15 molecule for doing a single stain control. So that's the problem here. Whereas if it beads, you can, no matter 15 or whatever, you tell me any CD molecule, no matter it is, if the fluorochrome matters, you can just add it to the beads, it will go and bind. So this is how we do. We never do it in beads because we have a good volume of the antibodies in different flavors, different fluorochromes, and we use cells to do compensation. So see how I have designed. So I am doing a compensation, single stain control compensation exercise for my 10 color instrument. Now, how I prepared is one tube only for FITC stained cells, one tube for PE stained cells, only PE, one tube for only ECD stained, similarly one tube for PC 5.5, PC 7, all single stain. Okay. Now you see how I've selected the markers. FITC, I've selected CD4. Okay. Now CD4 will bind to T helper cells. So the B cells and the other cells all will be negative, and the CD4 helper cells will show a good significant positivity, bright positivity. Then CD5. Now again, CD5 binds to T lymphocytes. So my B cells will act as a negative control, and CD5 will give you a good positive control, which is for PE. For ECD, I've taken CD8. Okay, for this tube, I've taken CD8. Again, your CD8 positive cells will act as a positive, and the other population will be negative. Then I've taken for PC 5.5 CD79B, which is my B lymphocyte marker. 19 I've taken for PC7, which is a B lymphocyte marker. 3 APC which is a beautiful marker for T lymphocyte. CD20 APC B cell. CD7 for T cell, CD2 for T cell, and CD45. CD45, when you are using it's a pan lymphocyte, you can add some unstained cells that will act as a noise. But again, lymphocytes are CD45 positive, as you know. See, I've used all the markers like FITC, PE for lymphocytes, markers against lymphocytes, right? So first I'll take lymphocytes, okay, MNCs or even the lysed WBC pellet, add CD4 FITC, run it in the machine, check for signals in other channels other than FITC. If there is you just need to give a compensation. You can do it automatically with the machine, or you can manually eliminate them. Okay. And then after finishing FITC, you run PE. Now you have taken cells stained with CD5PE, right? Run it in the machine, check for signal in other channels other than PE. Okay. If you get any signals in non-PE channels, then there is a spillover, eliminate them. You can automatically do it, or you can manually do it. So similarly, you need to run all the 10 tubes one by one. If you are using a 13 color flow cytometer and you are doing a 13 color experiment, then you need to do compensation with 13 single stain tubes. Okay, so it basically depends on how many colors you are using in your experiment, the compensation. Okay, so here it's a 10 color instrument, so we are using 10 single stain controls running, and then you do compensation. Okay, so that is about single stain control. So you run all the single stain controls, and then you can manually remove the spillover signals in other channels apart from the one which you have stained with, or you can automatically give the machine the instruction to compensate. It will compensate for you, right? Once it is all done, you can do a verification tube where you add cells stained with different markers and check whether there is any spillover happening. So that is how you do a single stain control. Now coming to fluorescent -1. Okay, so before I go into this, any doubts with single stain controls? Any doubts? Any doubts you have? If anyone have a doubt, you can just unmute and you can ask your question, please.
I guess no, because people in the chat have also said no.
Okay, right. That's great. So if there's no doubts, excellent. That's great. Great. So now we'll go into the second part, which is like, see, this is not really needed. I'm telling you, single stain control is mandatorily need to be done. Whereas the other concept is fluorescent -1, which is just reverse of single stain control. So can anyone tell me what is fluorescent -1? I'm telling you, this is not mandatory. Remember, single stain control is mandatory. Fluorescent -1 is not mandatory, but this is something reverse of single stain. So how do you do this? Assume that you want to do a FMO for FITC, okay? So which is fluorescent -1 for FITC. Now what you do, you take cells, stain with all the markers or fluorochromes other than FITC. In single stain, we were using only one fluorochrome and checking for spillover in other channels. Here it is completely opposite. You take cells, stain it with all other fluorochromes except the one where you need to check for the signal. Just reverse of it. Now you take cells. I want to check FMO for FITC channel. So I take cells, stain it with all other fluorochromes except FITC, run it, and I'll check for any signal in FITC. If there is a signal in FITC, it means like some other fluorochrome is giving signal in FITC, spilling over into FITC, right? So this is the other way of checking spillover. But now let me tell you, fluorescent -1 is not mandatory. It's not mandatory. You do this only for some very sensitive experiment where the separation between the negative and positive is very, very mild or very narrow. Okay, just to set your negative quadrant or negative cutoff, you may need to do fluorescent -1. For example, I'm doing an experiment where the CD25PE expression in lymphocytes. Now the CD25 in lymphocytes comes only in activated state and the separation may not be that great. But this is very sensitive and I need to check it perfectly. So what I do, I take a sample, stain it with all the fluorochromes except PE, run it, and I'll just draw the cutoff, making sure that nothing is beyond that. Okay, no other fluorochrome is giving signal in PE beyond that. Now I take the sample, stain it with CD25PE and run it. So anything beyond that boundary, which I've done with FMO, will be taken as positive, and anything within that will be taken as negative. Okay, so that's the concept with FMO. So I repeat, single stain control is mandatory. Okay, whereas in FMO is not mandatory, and you can use it only when there is a very sensitive experiment where you need to draw this negative cutoff perfectly.
I have a question.
Yes, please.
So is FMO is experiment specific? It's not done as
yes,
agent.
very
Okay.
perfect, bang on. You had the correct point. It is experiment specific. It is not like a general compensation. So it's not mandatorily done as a compensation experiment. It is experiment specific. For example, you are going to do a very sensitive experiment. You can do FMO to set your negative cutoff and then check with your experiment whether it's really positive or negative. Correct, you're right.
Dr. Thulasi, there's a question on the chat.
Yes.
Is it acceptable to generate a single 10-color compensation matrix and use it for all experiments, even those that utilize fewer than 10 colors? Or is it preferable to create experiment-specific compensation matrices? Example, one for TB NK panel and another for the lymphoma panel.
Excellent. Very good question. So let's answer this part by part. So you generate a 10-color compensation matrix for with the single stain controls, and you develop the matrix and you want to use it for all experiments. Yes, you can use it provided you are using the same fluorochromes, and without any change in the voltage of your detectors. That is very important. Okay, now coming to the second part of your question. So I've generated a compensation matrix for my 10-color machine. Can I use it for a 4-color experiment, 5-color? Yes, you can use it because you have compensated the spillovers across those fluorochromes. So even when you use a 4-color experiment, it doesn't gonna create any problem. I repeat, the two factors which you need to consider is, are you using the same fluorochromes which you have used as single stain control to do compensation first. Second, if there is a voltage change, then or your mission settings have changed, then you redo the compensation. You cannot use the old compensation matrix. I suppose I answered that. Right? So, right.
Yeah, you got a thank you for that, Dr. Thulasi. So
Perfect,
I guess
perfect.
Yeah, that should be good.
Right, super. So this is FMO. So for example, now, so what I'm doing is I'm just doing a FMO for P. So I've stained the cells. This is unstained where I've not added any fluorochrome. And here you can see that I've added all the fluorochromes except P. And I could see that if you compare with unstained, now you can see a signal here. Remember, I'm not added PE, but still there is some signal coming here, which may be from some other fluorochrome coming into the PE channel. Now, first with the unstained, I've set the negative threshold with the orange line. Now, I have taken up a bit more high, setting up the negative threshold with the blue line here. So this is the actual negative threshold with FMO. Now, for this sensitive experiment, when I run, now stained with PE, I can see the true signal here. This is the true positive. In case if I use the same orange line without doing FMO, I would have taken this entire as positive, which is wrong. Right? So that's the purpose of doing FMO. As rightly pointed out, it's more of an test specific, assay specific, not like a regular your single stain control. I repeat, single stain control is mandatory. FMO is test specific. If you want, you can do it. Now, how do you know whether have you correctly compensated? Okay, so here is a data where I could clearly see a FITC spillover into P, but how to compensate? So can I bring it down here? Can I bring it down here? Can I bring it down here? So which tells me that I've correctly done a compensation. For that, you need to use your statistics. So you have a software which has a great statistics. You need not calculate them, but if you just on a click of a button, you get the values. So the ideal way to say perfect compensation is done is, in this case, the PE median, when I say PE MFI, okay, PE median fluorescent intensity of FITC negative channel, negative population, and the FITC positive population should be same. I repeat, when you say the spillover is correctly compensated between two fluorochromes, in this case FITC and PE, here you need to check the FITC MFI. Remember, sorry, PE MFI for FITC negative and FITC positive population, it should be same. If it's same, then you say that you have adequately compensated. Okay, so
there's
you need to
question on the chat.
Yes.
Sorry to interrupt you, Dr. Thulasi.
No issues, please.
There's another question. What about if using the same fluorochromes, but the processing is different. Example, with cytoplasmic processing requires its own settings for comp.
Correct. I don't think so. Again, see, if you have adequately compensated, there should not be a spillover. So because we normally regularly use, when you do an adequate compensation, the processing until you know if there's a lot of debris and other things that may create some amount of signal here and there, but that is not because of spillover. Basically, as you know, it is because of debris. And a cytoplasmic processing, which includes your fix and permeabilization, doesn't gonna do a lot of change in that. Okay, so it doesn't, even the processing, if it changes to a minimal level, it doesn't matter. It doesn't gonna affect your compensation so much if you have done an adequate compensation with single stain control. No. Yeah. So that's how you need to check your whether the population have been adequately compensated. So I repeat, the spillover channel, in this case P, median MFI, which is median fluorescence intensity of the FITC negative population and the FITC positive population should be the same. When it is same, then you say it is adequately compensated. Okay, so if you really want to check perfectly, you need to do this way. But most of the practical flow cytometrists, they compensate, and when they say they are fairly okay, they go ahead. But if you want to make sure, you need to do this, right? So clues when you're analyzing the data. So as said, so though you have done a good compensation exercise, everything, but it's been a bit of time, say 6 months or 3 months down the lane, and there is some change in the instrument settings, voltages, and maybe some small things which will give you a hint that no, the compensation not adequately done, there is spillover happening. So what is that? So you can see this type of skewing or arrowing like effect, so you can see here that APC signal is spilling over into this channel, right? Similarly, you can see that this particular light is spilling over into this particular channel. So you can see this arrowing or a skewing effect, which tells that there is a spillover happening, right? So this is all some evidences to say during your data analysis to check whether there is a spillover happening. Now you can see here, can anyone tell me which is, you can see here, this one is adequately compensated because the PMFI for this particular population, which is FITC negative, and this particular population, which is FITC positive, is same. Okay. Whereas if you check here, this is undercompensated, which means like this has to be still brought down. Still there is a spillover happening. Okay. If you check the PMFI for this population will be more compared to this population, which means it is undercompensated. And you see here, this is over enthusiastic compensation, where you see that the PMFI for the FITC positive population is lower compared to the FITC negative population, which is called overcompensation. So again, these two are not adequate. This is fairly good. Right? Again, I'm showing you an example here. So which is adequately compensated, A, B, or C? This is adequately compensated. Okay. So you can check with the MFI, or normally we don't have time, right? During reporting, we adjust it to a level where it fairly looks okay. But theoretically, you need to check the MFI for the spin-over channels. Okay. And here this is overcompensation. This is undercompensation. Yeah. Sometimes even during our analysis, we may find data like this. So we cannot stop there, right? I know that there is some spillover happening to a minimal extent. If it is a very high extent, then we need to do our single stain control compensation exercise again. But if it is very minimal, what we do is we do something called post-acquisition compensation. This is done during our analysis. So what you do, you just adjust the compensation matrix or the compensation bar to a bit. See, this is this is uncompensated data. You can just compensate like this and bring it to normal levels. Okay. So this is, some people call this as naked eye compensation or cowboy compensation, but again, this is inevitable because though you have a perfect compensation matrix with single stain controls, very well done, but still there may be during your analysis, you may find small spillovers happening here and there, very small. You can just compensate with post-acquisition at the post-acquisition level. Okay, this is called post-acquisition compensation. Right. See here, you have a data like this. I can clearly see that there is FITC signal spilling over into PE. I can just adjust the compensation bar to a bit so that it comes down and it is adequately compensated. Okay. So, okay. Now, how do you know that we have got adequately compensated? Right. So is there any spillover happening? Because you cannot check that. The best way, remember, those who are practicing flow, make a n by n plot. What does this n by n plot means? Say, for example, if you are using 10 fluorochromes, make plots with all possible combinations. So if you are using, say, FitCP CD 10 fluorochromes, make plots across all fluorochrome combinations. So if there is any spillover, you can easily find out. For example, see, this is an n by n plot for a 8 color, correct? 1, 2, 3, 4, 5, 6, yeah, 8 color panel. Okay, now I made plots for every combination. So if you find CD56, it has, you can see plots with every other marker. Okay, now if you randomly see, can you able to spot out where are the problems with compensation? I can see there is some compensation error here that needs a bit of post-acquisition compensation. And what else? I can see some problem here, maybe slightly, and here there is some undercompensated. I need to compensate this to a bit. Even here you can see, so you can slowly adjust this, but very well compensated data, but still you can see some amount of undercompensation here and there, which can come in any sample. Though your compensation is very good with single stain controls, you can get this. You need to do it post acquisition compensation. You cannot be very strict saying like, no, I will not do post acquisition compensation and not everything on dot. That's not possible, right? So this is very important. This is very, very important. See, I'll just tell you one small story. We just some time back, this is some 5-6 years back, we taken a technician, okay, and we were standardizing one experiment. I could say it was like VASP for Wiskott-Aldrich syndrome. Actually, it was not giving signal. Okay, I was very, very disheartened. We were repeating many times. I was asking my technician to repeat it with multiple titrations, multiple processing methods, because I want to get this experiment done. The technician got very tired. It was a Saturday evening, and he was very tired. So what he did, like, he has tried 2 times in the morning. It has not come. The 3rd time also it didn't come. And 4th time I asked him to do it in a different method. So this guy got 4th time also it was not coming. So what he did, since the signal was not coming, he slowly increased the voltage of the channel. So when you increase the voltage of channel, what happens? Your PMTs are voltage driven. When you give too much of voltage to the PMTs, even a noise, which is a negative, can be pushed to the positive level. So he slowly increased the PMT voltage so that the negative came to the positive level. And then he said, like, sir, signal came. Then I saw, but still I caught that. During analysis, I found, because the negative population was also showing, the internal negative control was also showing signal. So I found like this is something gutbad, and I just abolished. And then he told me that increases voltage. Now, what is the problem here? Since he has increased the voltage, can I use the compensation matrix which I created earlier? No, that is gone now. That is gone. Once you have adjusted the voltage of the detectors, the compensation which you have done earlier goes null and void. You need to redo the compensation exercise. For forward scatter, side scatter, you can adjust the voltage and gains. That's not a problem. But fluorescent channel, if you have adjusted, it's done. You're gone. And before doing compensation, you should also adjust the voltage that the negative exit during the single stain controls. You can keep the negative population at the negative level. Okay, that also you can do. But remember, this is some take home. When you have done the compensation experiment exercise, compensation single stain control experiment, and you have set the compensation matrix once for all, don't ask your technician to touch on the voltages and gains. If he's going to do that, then your compensation becomes null and void. And when you run the experiment again, after some time, it may show a significant overlap and spillovers. So that's not the right way, right? So after doing compensation, you should not adjust the voltage. That's something very, very important. Okay. So during post-acquisition compensation, you may ask me, like post-acquisition compensation, you have done with the data. I mean, the acquisition file is done. So during analysis, you can change, you can adjust the compensation, but not during acquisition. Not definitely during acquisition, because if you change the voltages there in the detectors, then the compensation for the later part will not work. Okay, so now coming to the other important question, when to do compensation? This is something people ask very frequently, right? Should I do it monthly once? Should I do 3 months once, 6 months once? See, basically, it's like after your instrument setup. Whenever the instrument is being set up, IQ, OQ, PQ, the service engineer or the application specialist get this done for you. Okay, but don't always relying on them to do that. It's a very simple one. You people can, the technicians can also do that on a periodic basis. For example, if it's something which you design, say I'm going to do compensation every 3 months, great. You're going to do compensation every 2 months, fine. 6 months, it's okay. But anything beyond 6 months, or 1 year, like beyond 1 year, it's like you need to definitely do, because though you may claim that I've not adjusted the voltage, I've not changed my panel, but there is a lot of other factors which can slightly affect the detector efficiency, and still you need to do it. So a periodic basis of 6 months is fairly good. 1 year, okay, until your data is all good. Instrument breakdown. If there is an instrument breakdown, obviously the engineer would have worked on detectors, the voltages would have been adjusted. So definitely you need to redo the compensation exercise. Any change in the fluorochrome in the panel. For example, if you are using FITSI, and suddenly in another experiment you're using Alexa Fluor 488. Now that's a different fluorochrome. Or similarly, I can say PB in FL9, ninth channel, and you are replacing it with BB421, okay, which is also a violet dye. Now you cannot use the same compensation matrix what you created with Pacific Blue for brilliant violet dye. You need to change it, right? You need to change it. And when you're standardizing a new multicolor assay with a tandem dye, so again, a change in the tandem dye is very important. When you're changing a tandem dye, even that also is very important. You need to redo the compensation. So these are very, very important points when to do the compensation, but you are the right person to decide on it, because you see the data. Okay, you see the data, and when you see slowly the data quality is going down, and there is a lot of need for post-acquisition compensation you need to do for every experiment or during every analysis, then you need to say that it's time to do a compensation exercise with single stain controls.
I have a question here, please.
Yes, doctor. Yeah.
So if I change the fluorochrome, I have to change the compensation. This is okay. This is well understood. But I have to use the fluorochrome specific compensation.
Correct.
For this specific combination.
Yes, correct.
Okay.
So for example,
compensation metrics for different
exactly. Perfect. Okay. Perfect. So for example, if you have an experiment where you are using a different fluorochrome, you need to create a compensation matrix specifically for that particular experiment. So if you are using that, I mean fluorochrome for that experiment alone. For others, it's the conventional one. And if you are using an experiment where that only one experiment, if it's different from the others, you need to create a compensation matrix specifically for that. Yeah.
Thanks.
Thank you. So fine. So now the second part, I really feel, is it really needed? Because this is quite a tough topic, compensation. Yeah, I don't want to burden you with more of panel designing. So if you have any doubts, I would be happy to take. Panel designing, we can fix on some other day. Yeah.
Yes, Dr. Thulasi, with the interest of time, it was actually a wonderful session with the compensation itself. You went totally into depth of it. And we can always do the panel designing in such a way in another session, and that would be better, I think, for the forum if everybody agrees.
Yeah. Any
there's
doubts?
A question on the chat,
yes.
Vishesh Dudakia. It is assumed that manual composition should be done up to a limit. So how do we decide the limit to that? Kindly give an example.
Okay. Dr. Vishesh is there, so you can put me, I mean, I'll ask, like, there is I need some clarity on the question. So what do you mean by manual compensation? You mean to say the post-acquisition compensation, or during running the single stain control? Single stain control. I mean, your post-acquisition compensation.
No. Post-acquisition, yeah.
Right. So see, Dr. Vishesh, it's like, see, if you are just adjusting slightly, okay, across, again, there is no hard and fast rule here or a cutoff drawn here. So for example, if you are spending too much of time on post-acquisition compensation, then you need to do your compensation exercise, as simple as that. If say here and there some small adjustments, then it's fine if it's not affecting our time much. Okay, so there is no specific level to say you need to do a compensation exercise, but if it's taking a lot of time to set your data first before analysis, by doing post-acquisition compensation, you definitely need to consider doing a single stain control exercise to keep your compensation matrix correct. Yeah.
There's another comment on the chat. It's an iPhone.
Where this much percentage is acceptable.
Yeah, percentage, like so it's more of a percentage. I would say it's time. I would say that time. Like, say, for example, you work in a lab with a high load. No, for me, I cannot spend a lot of time on compensation. See, it's inevitable. Though you have a beautiful compensation matrix set, there may be a need to do a post-acquisition compensation, but you should not take so much of time. Say if you're spending half an hour time in setting your compensation, post-acquisition compensation, it's not the right.
Yeah.
No, maybe some 1 minute, 2 minutes just to check the data is all good and no, there is no compensation or skewing anything. Then fine, that's fine. Then you can just go ahead still. Yeah.
Oh, thank you. Thank you.
Thank you, Dr. Vishwesh. Yeah.
Thank you, Dr. Vishesh. There's another comment on the chat. It's an iPhone, so I don't know the name of the person. They said that it's a very nice class. Skewed curve is a spot that they had in their case. One of their cases. The skewed curve.
Okay. Yeah, skewed curve.
Is something they, I think, came across in their cases.
Yeah, then you need to check your compensation. So when you find skewing, okay, definitely there is a spillover happening. Okay, now the first thing what I would say for all the participants who are here, if you are really worried whether your experiment has a spillover and something, just go set up an n by n plot. I'll make it more clear. You set up all the dot plots across all combination of fluorochromes that's there in your panel. That will tell you which fluorochrome is spilling over into which channel. Set it right, or you can take a note and do a single stain control. Right? If it's a small one, then you can do post-acquisition compensation and you're really good. And if you find a lot of compensation across multiple channels, time to do your post-acquisition, single stain control compensation matrix, do it with it. Yeah.
They have said a thank you, clarity. Now they've got clarity and thank you very much.
Great. Nice. If everybody is very clear, I'm really happy because this topic is very tough. Even
definitely. It is very tough, but it is unfortunately the most important one. All interpretations depend on this. What is positive, what is negative, and without this is something that you cannot go ahead with the interpretation of the
Dr. Renu,
results.
I'll tell you some live scenarios. Some people send me plots and they come with the wrong diagnosis. But you will have a very rare finding. So they'll say sir, this is so-and-so case with expression of this, which is literally very rare. So I say okay, you don't send me the PDF, you send me the raw files. When they send the raw files and when you analyze, you will see a lot of spectral overlap and compensation issues, which they have not corrected and analyzed like that.
Right.
So you need to know the basics before you go for your analysis. That's very important.
Absolutely, just nailed that. Flow cytometry is such an interesting subject. It's the colorful part of hematology, I would say, when you're working in the flow lab, right?
I would normally say that flow is something like mathematics. People who love, mathematics, if you see, people who love mathematics will stick on to that. You will never bother about time. So flow is something like that. You love it, you will be there with it for long. You never see that as a work.
Absolutely. And it's very satisfying and gratifying, even though it takes a very long day in the lab. Sometimes we finish late into the night. But I think it's one of the most satisfying things that one can actually do.
And remember, everybody will be waiting for your verdict.
Oh, yes.
Yeah, the clinician will say, have you seen it? Have you seen it? What is it? What is it? The morphologist will wait whether the morphology is right. So so have you seen it? So that gives you, you are the decision maker. And remember, I'll tell you what, flow cytometry, you need to give your proper diagnosis. The people who are in clinical cytometry, okay, research is a great thing, but my knowledge on that is very limited. But clinical cytometry, if you are giving the diagnosis, when you are wrong, the other workup is all targeted on that diagnosis. And if you're giving a wrong diagnosis, the car goes on a wrong path, it's a disaster. So be very careful.
Yeah, so thank you so much for this enlightening session, which was taken really in depth. And I hope everybody here had a good time. And thank you for making it very interactive, especially to the audience. I would like to say that. Dr. Thulasi, can we have one last quiz question? So that, as promised to the audience, something, we could give back to them as well for their amazing interaction today.
Okay, some question. Right. So what should I ask is my thing. Okay, now, one thing which I would ask is, when do you run a unstained sample? Very simple question. When do you run a unstained sample? Yes.
You can put it up into the chat. You can raise your hands as well, and then you can come online. You can just turn your video and your audio on or audio on and give us the answer.
Yeah.
Take a
before
shot
running
at it,
actual
guys.
sample.
Okay. No, my question is, when you want to do an unstained sample, unstained means you take the cells no fluorochromes added, just run in the machine. For what purpose you check that?
Dr. Gautam is saying for APML
Internal
cases.
negative control also. Or autofluorescence control.
Autofluorescence. Okay, if you want to, so that is what Dr. Mohammed Bilal also saying. APML is known to show autofluorescence. The word is autofluorescence. So
okay,
when you want to check for so for autofluorescence, yes, there's a lot of people who are coming up now. Maybe it's a fastest finger.
Yeah. So was that Vishesh, Dr. Vishesh, who said that
he
answer
said
first?
that, yeah.
Okay, followed by Dr. Sendil.
A lot of
also.
people. Yeah,
a lot
great.
of people. So great, guys. I mean, I'm so happy that a lot of you thought about the right answer. We'll definitely see if we could do something for everybody who's given the answer here to encourage this even for the future. Thank you so much for the question, for on-spot question, Dr. Thulasi.
That was more difficult than giving the lecture.
Absolutely. But that was fun, and I think everybody would have had, rack their brains up a bit towards the end of it, right? Like to come up with that, put their thinking cap on, and come up with that answer. So that's indeed very great. Thank you so much again for everybody for being here with us this lovely evening, and especially on a busy day on a Friday to take time out from your practice to be here with us. Thoroughly enjoyed the session. Thoroughly enjoyed, Dr. Thulasi, for you being here with us today as always. And we look forward to the next session where we're going to look at panel design, and trust me, that's going to be even more interesting.
That will be definitely more interesting than this, because this is the more drier part. Yeah, definitely.
Now imagine this itself is so interesting how the next session is going to be. So we look forward to that. I myself am going to be looking forward to that. Get your questions ready for panel design even before you come for the session. We will be announcing that on the LinkedIn platform depending on the dates which are going to be convenient for Dr. Thulasi, and we'll keep you posted on that. And Mr. Manohar Dhaka, thank you, Dhaka, for being on the call today and making this happen. Without you, this Zoom call would not have happened today. And I'd be very happy if you could reach out to Dhaka, and Dhaka will reach out to you for the prizes. So we'll be in touch with all of you, and yes, we'll keep you posted on the following developments in this front. Thank you again, and have a great evening.
Yeah, thank you so much. Bye.
Thank you, Dr. Thulasi, again.
Yeah.